everyone. This is Suhas, Assistant Professor, Department of Pharmaceutics. So today we are going to discuss about the topic phytosomes. So what are phytosomes? Let's see first the contents what we are going to discuss in this particular chapter. The introduction to phytosomes. What is the structure of phytosomes? Properties of phytosomes. Structure of phytosomes versus liposomes. How does it differentiate? Advantages of phytosomes, disadvantages, preparation of phytosomes, evaluation parameters, application of phytosomes, and some of the marketed products that are currently available, and also the reference which is cited. So introduction. So what exactly phytosomes means? The phyto means plant. As you all know, phyto means plant or plant extract. Plant extract. Zome means cell-like structures. So phytosomes is a novel drug delivery system that combines hydrophilic bioactive constituents of herbs or herbal extracts, which is bound by phospholipids. That means by creating phytosomes, you can achieve more bioavailability. They like more bioavailable than a simple or conventional due to its enhanced capacity to cross the rich biomembranes and reach circulation. That means you know the body or the skin has a lipid bilayer, right? Also, anything if you want to give it through your skin, then it has to pass the lipid bilayers. So better absorbed and better. So applied to standardized plant extracts or water-soluble phytoconstituents and many popular herbal extracts including grape, olive fruits, leaves, green tea, ginseng, etc. into phospholipids to produce lipid-compatible molecular complexes. So most of the bioactive constituents are water-soluble compounds like flavonoids and glycosides. See, the major issue or drawback which we face delivering of a phytoconstituent. That means they are mainly water soluble drugs, and these water soluble drugs has to be properly absorbed. So some of the other disadvantages like the denaturing of your phytoconstituents. So you have to find a new drug delivery system that is which is phytosomes. So because of their water soluble property and lipophilic outer layer, it shows better absorption and produce better bioavailability. So the presence of a surfactant, that is the phospholipids in the molecule, these are shielded from water triggered degradations. So there will be no degradation of your dosage forms. So molecules are anchored through chemical bonds. So these are anchored through chemical bonds and polar chain of the head of the phosphatidylcholine. It can be demonstrated by specific spectroscopical techniques. So this is the structure of a phytosome. So as you know, phytosome contains phospholipid, whereas curcuminoid is our phytoconstituent. So what has happened is there is a complex formation between the phospholipid and the phytoconstituent. So this complex formation where you are hydrophilic head of the phospholipid. So a phospholipid contains two nature, the tail of hydrophobic nature and the head of a hydrophilic nature. So curcuminoid is forming a complex between phospholipid becoming a curcuminoid phospholipid complex. So properties of phytosomes. Chemical properties. The first property we study is chemical properties. So phytosomes is a complex between a natural product and natural phospholipids. So the phytosome complex is obtained by reaction of suitable amounts of phospholipid and the substrate in an appropriate solvent such as glycerol. So the main phospholipid substrate interaction is due to the formation of hydrogen bonds between the polar heads. So as I said, the phospholipid has a head which is hydrophilic. So there is a formation of a substrate or a complex between a phospholipid and a chemical substance, that is phytochemical constituents. So that forms an hydrogen bond. So this substrate complex is more stable when compared to other dosage forms. So, and the polar functionalities of the substrate. When treated with water, phytosomes assumes a micellar shape forming a liposomal-like structures. That means a circular zomal structure where a water molecule, in the sense hydrophilic drug molecule, is covered within the phospholipid layers. Biological properties. So phytosomes are advanced forms of herbal products that are better absorbed, utilized, and as a result produce better results than conventional herbal extracts. Why it is showing more response compared to conventional drug means drug extracts or drug formulations means, see, a phytosome is a water soluble drug. You all know you have to pass a lipid bilayer. So if you want to pass a lipid bilayer, the outer layer has to penetrate through that lipid bilayer. If you are placing anything which is soluble in water, it does not penetrate through your lipid bilayer. So phytosomes are lipophilic substance with definite melting point, freely soluble in nonpolar solvents and moderately soluble in fats. So phytosomes can accommodate the active principle that is anchored to the polar head of the phospholipids, which finally becomes an integral part of the membrane. So the increased bioavailability of phytosomes over the non-complex set botanical derivatives has been demonstrated by pharmacokinetic studies and pharmacodynamic studies. As you know, pharmacokinetic studies and dynamic studies means when a drug is administered to the body, what the drug is doing to the body and what body is doing to your drug. So that is pharmacokinetic and pharmacodynamic properties. So next, let us know the difference between the phytosome and the liposome. Why it is known as phytosome, why it is liposome. See, as in phytosome, the active chemical constant molecules are anchored through chemical bonds to the polar heads of the phospholipids. Whereas in your liposomes, the active principle is dissolved in the medium of cavity or in the layer of membrane. So there is no formation of any chemical bonds. So in phytosomes, phosphatidylcholine and plant compounds form 1:1 or 2:1 complex depending on substrate. Whereas in these hundreds or thousands of phosphatidylcholine substances or molecules surround the water soluble molecule. So phytosomes much better absorbed than liposomes, showing better bioavailability, whereas bioavailability of liposomes is less than the phytosomes. Contents of phospholipids is less when in phytosomes, where it is higher in your liposomes. So this is how you differentiate in the structure. See, in A you can see there is a complex formation between the phospholipid and the drug, which is a plant extract. See, that is in liposomes, the drug extract is at the center or in the where phospholipids is covering. So this is the major difference between liposome and phytosome. Advantages of phytosomes: it enhances the absorption of lipid insoluble polar phytoconstituents through oral as well as topical route, showing better bioavailability. As the absorption of active constituents is improved, its dose requirement is also reduced. So when there is more bioavailability, then you can decrease the dose which has to be given to a particular patient or a. So phosphatidylcholine used in preparation of phytosomes, besides acting as a carrier, also acts as a hepatoprotective, hence giving the synergistic effect when hepatoprotective are employed. So as you know, phosphatidylcholine also acts as a hepatoprotective. Synergistic effect means it is giving a benefited effect with your drug molecule which you are administering. So chemical bonds are found between phosphatidylcholine molecule and phytoconstituent. So the phytosomes show better bioavailability or better stability profile. So added nutritional benefits of It assures proper delivery of drug to the respective tissues. The nutrient safety of the herbal extracts need not be compromised by conveying the herbal drug as means of phytosomes. So entrapment efficacy is high and more over predetermined because drug conjugation with lipids in forming vesicles. So there is no problem in drug entrapment while formulating phytosomes. So phytosomes are also superior to liposomes in skincare products. The major disadvantages of phytosome is phytoconstant is quickly eradicated or eliminated from the so the major drawback is it can be eradicated because when the water molecule it can be of a danger of oxidation, any other denaturing reactions. How do you prepare a phytosomes? The general method is take a phospholipid, that is phosphatidylcholine, dissolve it in an organic solvent where your drug is dissolved, usually like methanol, ethanol, alcoholic extracts. So solution of phospholipid in organic solvent with drug extract is then evaporated. After the evaporation, there is a formation of film. That after evaporation, there is a uh, PPT, or you can call it as a thin film which has to be hydrated. So this hydration leads to the formation of phytosomal suspensions. So the next method is anti-solvent precipitation technique, whereas drug plus soya lecithin is used. Reflex with 20 ml of dichloromethane at 60 degree for 2 hours. Then concentrate the mixture to 5 to 10 ml. From that 5 to 10 ml, for that 5 to 10 ml, add hexane 20 ml. So filter the PPT found, dry, crush and pass through mesh size 100. So rotary evaporation technique. So use a rota evaporator where drug and soil lecithin is dissolved in 30 ml of tetrahydrofuran, stirring for 3 hours at a temperature not exceeding 40 degrees Celsius. So thin film is formed. So this thin film is mixed with N-hexane with stirring. Then precipitation is obtained. Dry and pass through mesh, the phytosomes are formed. Solvent evaporation method. As previously we have discussed, drug and soil lecithin reflects with 20 ml of acetone at a temperature of 50 to 60 degrees Celsius for 2 hours. Concentrate the mixture to 5 to 10 ml. Obtain the precipitate, filter and collect. The phytosomes are obtained. Different additives used in the formulation of phytosomes. So phospholipids, soya phosphatidylcholine, example phosphatidylcholine, egg phosphatidylcholine, dipalmityl phosphatidylcholine itself, and also phosphatidylethylamine. Aprotic solvent like dioxane, acetone, methylene chloride, non-solvent like N-hexane, alcohol substance like ethanol or methanol. Evaluation parameters, one is transition temperature. The transition temperature of the vesicular lipid system can be settled via differential scanning calorimetry. So if you perform a DSC test, then you will know what is the transition temperature of the vesicular lipid systems. Entrapment efficacy, the entrapment efficacy of phytosomal preparation can be determined by exposing the preparation to ultracentrifugation. From that you collect a supernatant liquid, test for the drug or molecule or the amount of drug molecule that is present. So then you can have a comparison with the amount that you added and the amount which is entrapped in your vesicular trabecular system. So vesicle size and zeta potential. The particle size and zeta potential of phytosomes can be confirmed by dynamic light scattering, which uses the computerized examination system and photon correlation spectroscopy. Surface tension activity can be measured. The surface tension activity of the drug in aqueous solution can be determined by Dunoyer ring tensiometry. So visualization. Usually, what is the size, what is the shape, and how does it look? Whether it has a vascular uh, shape, what kind of shape it is there. So you can check it through SEM. Sometimes STEM also can be used. Vesicle stability. The steadiness of vesicles can be measured by calculating the size and structure of the vesicles over time. The mean size is calculated by DLS and structural changes are monitored by TEM. Spectroscopic evaluation to confirm the formation of complex or to study the reciprocal interaction between the phytoconstans and the phospholipid H1 NMR or C13 NMR of FTIR are used. In vitro and in vivo evaluations, models of in vitro and in vivo evaluations are selected on the basis of the expected therapeutic activity of biologically active phytoconstants that are present in the phytosomes. So application, silmarine phytosomes. You can prepare a phytosome of silmarine, phytosomes of grape seeds, phytosomes of green tea, marketed products, that is silicon phytosomes, which we saw in the previous slide, grape seeds phytosomes, hawthorn phytosomes, etc. So this is about the phytosomes, and we have studied what is the structure of phytosomes, what is the chemical nature, the biological nature, how it is prepared, and where it is used. Thank you.